Hello, I'm Massimo Pernice. I'm working at ICM, Institute of Marine Science in Barcelona, as a postdoc, and I'm also in charge of the flow cytometry lab. And today we will talk about flow cytometry application in microbiology. I'm a microbial ecologist, so it will be more about ocean and sea and this kind of stuff. This is the title that I provide, and this is the fancy title that I prefer. That is a little bit more funny. And let's start. Flow cytometry is a technique that allows us to answer two questions: who and how many. And basically, we count bacteria, algae, and other microorganisms thanks to their optical property. We always start with a sample that is fixed or fresh. This makes a little bit of difference in the lecture, but not so much. And we have always our flow cytometer that just looks like a box. Today we will try to see what is outside the box, inside the box. And inside the box, we have the flow. So basically, the machine took a liquid that is named sheet fluid. So it makes two little sheets to squeeze the sample and to have one cell of our sample that passes in front of a laser. Once the cell passes in front of the laser, it emits fluorescence, and we collect in graphs like that are named cytograms. So it's really important that just one cell pass in front of the laser, because if we have more, of course, we don't have a proper measure. This machine was developed at the beginning to work with blood and things like that, and we adapted with time to working with the marine samples. So it's no one thought, oh, let's do this for marine biology. And once the cell passes in front of the laser, it's excited, and so the information is collected by several filters of different color. Let's take a better look. We have a property that is named scatters. This is not fluorescence, it's how the light is deformed by your sample. Normally we have forward scatter. This is the scatter. If a cell is little like that, the light will divide like that. If it's big, it will be like that. So we can relate this property with the size of the cell. The more the cell is bigger, the more it will be bigger the scatter. We also have the side scatter, so how much light, blue light, go on the side in an angle that is from 90 degrees to less. And this information is more related with morphology and granulometry, and also with size. We saw that when we work with bacteria, this is better to characterize the population. And all these things that I will comment today, we will see later better at the machine because it's really more easy to see it doing. And after that, we have the fluorescence. There are two types of fluorescence. This an inner fluorescence of the sample. This is the most normal example is the chlorophyll A. So normally we have we know the excitation, in this case in blue light, and the emission in red light. And we have a filter that is represented by this rectangle. So the filter will collect just this part of the emission of fluorescence. We can also use stain that provide fluorescence to our sample. The one that we will use today is cyber green, but it is orange if you look at it, but it emits in green. And in this channel that is called FITS. And cyber green is a DNA stain. This means that it will stain also your DNA. So we always manage this with the gloves because you can have cancer with this. And the important thing is that the quantity of cyber green is proportional to the quantity of DNA and RNA of RNA and DNA of the cell. If you work with the microscopy, you have to understand that flow cytometry is really, really similar to microscopy, but the only difference is the work is done by the machine. But if something is green in the microscopy, you will read in green in the flow cytometry. Or if you see chlorophyll in the microscope is really red, and you will look for the red in flow cytometry. So I have a lot of students that came to the lab and say, I don't understand all this dot, all these things, but it basically microscopy, but in another way, you know. And normally the results are in this graph that we call a cytogram. As you can see, this is the same name of the channel. Every cytometer has different names. Sometimes you just see filter 1, filter 2, filter 3. And here we have the scatter. In this graph, each dot is one cell, in this case, bacteria. The last thing of this stain. This stain is not specific for bacteria. It's specific for DNA. So it will stain phytoplankton, bacteria, fungi, viruses. So it's the operator that has to know the sample to understand exactly what it's doing. You can also see that there is a little h near the name. So the sample in general, we can read it like weight of this peak of area of the peak. There are little difference, but they are not so important. Just to I explain this just for you to understand when you see this letter. Sometimes the area, you know, has a little second peak, so they are not completely proportional. And also the number of the axes are non-dimensional because this dot can move if we give more voltage to the machine. So it's not, you cannot think about flow cytometry like spectrophotometry. It's not that we say, oh, this is the quantity of chlorophyll, or this is, in this case, the size. No, it just, it moves, it's relative. We know that something here is bigger than something here, or something here has more DNA than something here, but it's just a relative measure. There are ways to make it not relative, but in general, it's relative. And finally, how to decide what is a population. So we have a lot of dots. Sometimes it's really clear because you see a space between populations. So you say, oh, this is something and this is something else. But it's also not always like that. There are two philosophies when the people make gate to leave a fixed gate always in the same position and just pass your sample and say, oh, this is what I want to count. Sometimes people just go one by one and do it like that. And also, normally we do things manually because you have a student and you ask to the student to do it. Now there are a lot of automated methods in Cytoflex, the cytometry that we will see today. You just can click automatic and it will make the gate. Otherwise, there are scripts with R or I guess also with Python to just say, can you please understand what is a population? And normally the results are expressed by the machine like event per microliter. And we normally in microbial ecology use cell per milliliter. We will see this also at the machine later. And this is the machine that we will use today, I hope. It's Cytoflex. Cytoflex, the one that they have at the university is really good because it has 7 lasers. You can see the laser here. And these are all the detectors the machine should have. So normally when you take the manual of the machine, you can understand how many colors can you use. We normally use only the fluorescence from red laser and blue laser, you see. For example, the cyber green need the blue laser to be excited and emit in green. And here you have the name of the channel, Fitch. But for example, people in biomedicine, they use a lot of probe, for example, when they want to study cancer and the different type of population of in the blood of a person with cancer, they need more color to characterize different population. And when you have more color, of course, you have to be sure that this color doesn't overlap. And for this reason, we use the cyber green because in nature, we always have chlorophyll. So we need another color that doesn't overlap a lot with this one. I put this, I think you have also in the PDF. It's a spectrum viewer. So when you have to choose a color, for some reason, it's a good way and really fast way to say, oh, this doesn't overlap. And you can buy your stain or your product. And a little bit of characteristic of the Cytoflex. I'm really happy with this machine. I don't sell it, but I really think that it's kind of the Ferrari of the flow cytometry in this moment, because every university and center is buying the same machine because it really works well. The software is also really fine, and compared to the machine that there were before, it's amazing. There are a lot of laser and filters, as I said before. You have a max of 10,000 events per second for reading. So I say that you have to have just one cell. If you pass this threshold, you will have more than one cell in front of the laser. This means that when you work, you don't have to dilute, which seems a really stupid thing, but it changed your day, And before it was like 1,000, so it's really good improvement. It doesn't work under pressure, so the flow is really regular. Before, you really have to look at the water that have to be always at the same level. It makes the vortex by itself, which is really nice. And it has violet scatter. What I commented before was the blue scatter, which is the normal scatter of the flow cytometer. Violet has a narrow wavelength, so it's better to characterize really little population. Now there are a lot of people that work with viruses or vesicles, so it's a really nice things that this cytometer have this scatter, and you have just to mix to filter to have it. So it's quite easy and useful. And let's start to what kind of population we can measure with this machine. We will start with phytoplankton because was one of the first things that people start to do, because phytoplankton has a lot of autofluorescence. Normally, in a normal marine coastal sample, we saw 4 populations that fluoresce in red, or in some cytometry is named filter 3. So we have Pleurococcus, Synechococcus. These are both 2 photosynthetic bacteria. And after that, we have pico-eukaryote and nano-eukaryote. Bigger things like diatoms sometimes are really on the top. There is no resolution to see diatom, and they are too much big for this machine. But there are machines that are more specific for bigger organisms. One thing, when we say pico-eukaryote and nano-eukaryote, is a convention. It's not the normal sites that we intend pico and nano. We just saw two population, and we say pico and nano. Now the tendency is to say eukaryotes, and that's it, because you really don't know what is there. This is an important thing of flow cytometry, is to understand that it's a rough technique. You never go really into this. It's not like omics. You cannot say, oh, this is this specific group. But I think it's still really useful. Normally, we identify this thanks to Synechococcus, because it's the only one that has fluorescence in yellow. We will see later this also to the machine. And Pleurococcus is always the one under Synechococcus, and we know that eukaryotes are on top of that. At the beginning, they just have culture, so they know where everything goes in the graph. But if you really want to know what is something, you should use sorting, which is a flow cytometer that can select your population, and after that you can took your sample and go to omics and say, oh, this is this species. So you always have a little bit of you need a little bit of knowledge of your sample to understand what is there. Otherwise, everything is just bacteria or eukaryote. And this is a sample of bacteria stained with the cyber green. As you can see, here we have the forward scatter, here we have the green, and all these are bacteria. I put a little bit of bits here, which is this blue population there. These are little dots that we use first to see that the machine is working well, and also to have a really rough estimation of the size. In this case, I think these bits were 1 micrometer, so we know that all these things are less than 1 micrometer. We can also use this to more or less know the diameter of the bacteria, because we know the value here And so we can do a proportion. In this other graph, it's the same sample, but you can see that it's green versus red. So sorry for the misleading, because I put the photosynthetic one in green, but these are the one that fluoresces more in red. And you can see that they really pop up from the rest of population. For us, it's really important to define what is photosynthetic, because normally you from the same sample, you do phytoplankton, and you see the photosynthetic part without compromising the sample. And after that, you want to know something about the no photosynthetic part. So we use it basically to put it away and say, oh, I'm not interested in this moment. An interesting thing here is that we don't expect this population to be here, because this population shouldn't have red. And actually, if you take a look, I saw that it's really good to use cyber green, because there is no overlap with the chlorophyll, but there is a little bit of overlap. This is a really little bit, but it counts, because you expect your populations without chlorophyll to be here. But this little overlap to make that the filter red took information also from the cyber green, and the population go in diagonal. We are not worried about that, because the things that are chlorophyll really go out this, but normally people apply a thing that is named compensation, which is the correction of the fluorescence spillover. So normally you just put green bead in the machine, and you say, I want this green fluorescence. Just take this green fluorescence. And so mathematically, the machine will put this population from here to here. We will not do this today, because in microbial ecology, we realize that we don't need it, and we prefer to not do it, because if you can work less, you don't do it. And another thing about prokaryote is that we always say two population in a marine system: high nucleic acidic bacteria and low nucleic acidic bacteria. I want to highlight that this happens in the marine environment. So you can always, it's not always true. For example, in Prodigio sample was not true at all. You have a lot of population, but coastal environment are kind of simple. And these two populations are, here you have two different samples. You can see that here you have a lot of high DNA contact bacteria. This is the low, they are less. And here you have a lot of low. So we assume that the high, since the stain is proportional, are more active, because they have more RNA, and maybe they are duplicate the DNA, so they are in phase of duplication. So we can use the ratio of high nucleic acid content versus low to have some information about the activity. Was in some paper, they see that this ratio is proportional to the bacterial production, which we measure with radioactive leucine. And so we believe in this, but still there are little things that never will be big. Even when they are really active, they will not go so up. So it's, again, it's useful, but it's not perfect. And another technique that I want to show you, because it's quite common, is the use of two stains, one for green and one for red with the propidium. And with this technique, we can establish the number of dead cells. So the propidium just go inside cells that have the membrane damage. So we can use, we stain the same sample with these two colors. And what happens is that if you see in red, the damaged cell will pop up from the rest of population, and you can make a ratio. This red is different from the red of chlorophyll. It's far red. It's a little bit moved. Again, there are critics also on this, because sometimes the permeability of the cell is changed with the physiology of the cell. But also in this case, we see that there was a proportionality with the bacteria production. So we keep doing and believe it. Now, you can do this only with fresh sample, of course, because if you fix, everything is damaged, so the propidium will stay in the entire population. And we also can measure viruses, basically with cybergreen, but in this case, it's diluted in milliQ, and the sample is really more diluted than for bacteria. Viruses are also always on the left of the bacteria and top down, bottom down. And you will see that there are always like line of different quantity of DNA. Normally, we believe that the one that are with more DNA are viruses of eukaryote, and this part here near the garbage are viruses of bacteria. And this is, well, it's not so recent, but it's the last thing that we add to the toolkit. And I recently worked with fungi, but not in flow cytometer. And there are stains that we still didn't try in flow cytometry. We try only in microscopy that are specific for fungi, because fungi has chitin, and we have the chalcofluor. This is quite common. And also what we used here, which is in this red picture, is the wet germ agglutinin. And in this case was conjugated with Alexa 594. Of course, in this case, we were working with a culture of fungi. So we have here our DAPI, which is in just the nucleus. Here we have our probe because we were testing a probe that was in green. So we decided to have the same for the chitin in red. But if you work with a sample from, for example, an algae culture, probably you have to choose another color because there is no point to have red and red. It's really complicated for the eyes. But this is just to know that there are also a specific stain for the eukaryote. If you want to characterize, for example, protease flagellate and this kind of stuff, where the topic is that no one just sit and say, oh, I have to make a protocol for this for now, because there are a lot of things to do, and no one puts his energy in technique because it's time consuming. But there are ways to count. Also, people are still really used to rely a lot on microscopy. They don't like to. It seems like still too much flow cytometer, but things are changing. And now I will show you a little bit of data. This is a paper, is methodological paper that I published last year about automated flow cytometry. It's in the Mediterranean Sea. So I use this cytometer. It's different. It's Akuri. This is my face because I was desperate. Nothing was working. And this is the boat that we used, the Garcia del Cid, that was just dismissed this year. I think this was one of the last cruise that he did. So I took seawater from a tap, and that was connected to a pump that was at 5 meters, so I can do the surface. And this machine that we will see later better, it's an automated sampler. So I have a sample of bacteria each 15 minutes. So this is the machine. It's named OnSite. I think it has been developed recently from a startup in Switzerland. So basically, this machine, it's more like that. This is the nice one. So it takes sample from one side, and all this other tube is just to clean itself. So you take the sample, you stain it with cymer green in the very same way that you do with your eyes, and just move the sample to the cytometer. This is really useful to have a time resolution really narrow, which is for us is amazing. And you have a syringe here, which is the one that makes the job. So this is our results. Each dot is abundance of bacteria, like cell per milliliter. Here we have Mallorca, here is Barcelona. So the cruise was, we are always in the center and go like a flower, going back, like this. And you can see that you can really see different color. It's when it's violet is few bacteria, when it's reddish is more bacteria. So you can see that in the center we have the green. When you move till the coast, it became blue, and when you move till Majorca, it became yellow. So we have higher value of abundance near Majorca. We have just this really strong abundance near the port there because we entered there, and the port is really a mess. And here you can see just the abundance of the station. So this is the central station. It was a little bit constant, but go down during the time. And every time that, again, we go to the coast, it was less than in the middle. This day we have a storm. I always cited this because it was really nice that the machine keep working during the storm. So for me, it was perfect because I go to the bed because I don't want to throw up, but the machine keep it and was like, and then the range of the data was so narrow that I know that it worked good because there was nothing that say what the machine had done, you know, and this was really nice. And this was the first time that we used the machine. We are really happy with that. And also I pulled together all the day sample and all the night sample because you can also we see the information through space here, but you can see the information through time. And again, the abundance of bacteria during the day is slightly higher than the abundance of bacteria during the night. And another interesting thing is that this is still abundance of bacteria. So we go from station D to station S. We stay 9 hours here and go back. And this day was, you can see that there were really two water masses because when we go back after 9 hours, we go back to the abundance of before. The interesting thing is that this is the salinity of the area. So you can really know that this were two different water masses. And this happened also with when we go more near Mallorca. So it really doesn't change during the days. And we spend 3 hours here. Was really interesting because when you measure, you never go back to one place and you don't know, and you just believe that your technique is working. But we really saw how much flow cytometry is. It's rough, but it's powerful because it's you have a real data. And this is the arrival at the port. This is the last hour. It was really nice to see how much the port is dirty. Then terrible. And this is another set of data. This is not published yet. I went to Antarctica in 2000, well, 2 years ago. And the Antarctica is really important environment in the Earth. It influences the global climate and the global ecosystem. And in particular, the microbial community is really important for the primary production because a lot of the global food web is based on the plankton of the Antarctica, and also is really important for the carbon sequestration. They calculated that 30% of global ocean uptake is taken in the Antarctica. So we found it really interesting to have a huge cover of this area, and we take sample for bacteria or for phytoplankton. This is a picture of the salinity and how much it varies along our cruise. We start on the top, which really huge value of salinity is 34.5. And it's go down and down because it's found more sweet water. On the offshore, it's a little bit more stable. This is the Hesperides. It's a military boat that we can use in Spain. And this is a real picture from the cruise. We go on land, and it was really nice. So the cruise lasts 2 weeks. So I have 2 weeks of sampling, and we took also temperature and salinity around the entire cruise. And so here I will show you just a little bit of the value of abundance, because I don't want to bore you, but this area was the beginning of the cruise. I don't want, we start and finish here. This is Deception Island. It's named like that because they think to found a huge island, but was empty inside. And you can see that the bacteria abundance really go up here and here. And the interesting thing is that both here and here, we have human presence because the Spanish base is in this island. Here we have volcanic activity, which means a lot of nutrients. We also have penguins and sea lion, which means a lot of shit, which is food for microbial. And also there is this horseshoe shape that make the water really less in communication with the outside. And this makes really the abundance go up. So again, flow cytometry is, you can see that geographically these data are really a strong meaning. So again, flow cytometry is really powerful to establish the bacterial abundance, and especially in an automatic way, because you can took all this sample, but it's really complicated. And as I said before, we can use the forward scatter to have an idea of the relative size. So you can see that the sides change a little bit during the cruise, especially here in the south part, and we have a peak of huge cell here in Margaret Bay. And the size is a little bit more higher. There is more green here than here. The interesting thing is that if we took the offshore water, so all this sample, you can see that there is a really good relationship between the relative size and the abundance, which is expected because, you know, the K and R species, you know, so we have a lot of species that are really little and few species that are really big. This relationship is broken in the inshore water, probably because there is so much predation of flagellate and things like that, that there is no time to see the relationship, but probably it's still there. And as I said before, we can so to the low DNA content bacteria and high DNA content bacteria, since these are the two main groups, the two graphs mirroring each other, because when one go up, the other one go down when you do. And I did it with, in this case, with bioinformatics, with a soft script from Flosson, is there. And we can use this abundance to calculate the activity of bacteria. And again, the activity change around the coast. It's a little bit big in this area and really huge here in the south. We can also use, take a look to the phytoplankton. In particular, I want to focus on this group. This group is cryptophyte. They have a lot of fluorescence in yellow, but we are sure that they are not Synechococcus, because Synechococcus is not in the Antarctic at all, and also they are really big. The important thing of the so where it's completely absent in the offshore water and is present only here. The important thing of this group is that if this group grow, it displays diatoms. So it changes the entire structure of a trophic web. So if there are more cryptophytes, like in this case, there will be more salps and less krills. So if we have less krills, this is a problem for whale naturally, because the whale eat the krill. So just with the, again, the flow cytometry is really powerful because we can have insight on the entire trophic web just with a normal sample. And it was really nice to discover that. We were also lucky because we have a story now. And And now this is just to know that exists. There are, if you think about the fluorescence as I talk about it, it can be conceptualized as phenotypic characteristic because you know, if there is more fluorescence in red, there is more chlorophyll. If more fluorescence in green, there is more DNA. We also talk about the scatter, so it could be more bigger, more. So all these characteristics is like if you have an animal and say, oh, this animal has 4 legs and or 3 eyes, whatever. So we can use this characteristic to calculate a diversity that in this case is a phenotypic diversity. And from all these characteristics can be put in a table. And from this table, we calculate for each group of cell this diversity. This is all these techniques now are grow and grow. They go under the name of computational cytometry. It's something like what happened in the last 10 years with omics. So now there are a lot of people that are developing new scripts and new tools to make this calculation, both to identify group and to calculate diversity. So the diversity calculated for this set of sample, it was really nice because it goes from blue, green, and red. And it was surprisingly proportional with salinity. So salinity will explain the variability of alpha diversity in this case. These dots, well, they explain me later that they didn't put on the machine, so they are there, but they are not real. And yeah, and this is the salinity. So you can see that the change of the salinity really mirror the change of our sample. So of course, the salinity changed because there is the global warming, and this global warming really affect the microbial community. So the interesting things is that we can catch this change with flow cytometry. So in general, every change in parameter like temperature and salinity is reflected in the community and it easily detected with flow cytometry. So I was really fast or what?
No.
Okay.
20 minutes, but then we have time for questions.
Okay. So from one sample, we have information about the total abundance, group abundance, because you can see high DNA bacteria, low DNA bacteria, photosynthetic bacteria, the relative size, the biomass. So if we can calculate the diameter of each cell, there are equations that can change the volume of this cell in biomass. It's not perfect, but I mean, the equation of the carbon is really good, but there we are still adjusting how to calculate the diameter of the cell because we use the beads and the light really trespass the beads in a different way because cells are more translucent. So when you see this is less than 1 micron, is not exactly true. We can have information about the activity doing the ratio of high and low. And we can also now have information about the diversity. This diversity, I forgot to say that they say empirically that it's not equal to the diversity that we calculate with 16S, but the pattern is the same. So we still believe that it works. And that's it.
Okay, thank you. Thank you, Maximo, for the excellent presentation. Very well presented and very dynamic. We have time for questions now. Yes, Belén.
Check the bacteria cells are alive for that can also be used for the microalgae.
I think you can use it for the microalgae. I never did it just on the sample of microalgae, but I think there is no problem.
And the other, the cytometry group is interesting to combine the omics.
Yeah, so I just sailed from Vigo to Canary, and we took sample to make the extraction. So I took the sample from the same tap water, and we want to back check because when someone want to sell you the technique, say it worked, but we are not sure 100%, is quite new. So we want to sequence the 16S and make the diversity with cytometry and see if more or less is the same. It shouldn't, because flow cytometry is a phenotypic diversity, and it has also the information of the physiological state of the cell. So if a bacteria is in division, it will go up in the graph and will look like another population. But for 16S, it will be the same, because the 16S is the same if the cell is dormant or dividing. So this is basically it's analysis of images. You will see how much dots are in the same area. So we expect the same pattern, but it not has to be 100% equal. But the important thing that you can see where the change happen, and maybe if you have a lot of sample but you don't have money and you want to decide where should I sequence to see difference, you can use the diversity from flow cytometry to say, oh, here and here I'm sure that it will be different. Like for example, the one that we see from with the bot, there is no point to do 10 point here and 10 point here. You can do 1 and 1.
More questions, Daniel?
For the presentation. You mentioned that it is a Ferrari, and I agree, it's a very nice instrument, but it's also a very concentrated.
Yeah.
Do you see any efforts for making it simpler and more and cheaper for other applications instead of?
No. No, we have the other one, the one that I use on the boat. It's really cheap. It's 4,000 euro. It's, I mean, cheap, scientific cheap. But it's really broke continuously, and
Ones are very expensive.
the good one is really expensive, and they are really. I mean, if you want the sorter is 10,000.
They can, they also can, right?
Yeah, yeah. But I think it's good investment. It's also sometimes you feel better with the machine just because the software is it works better, and I always have to also teach to students how to use it, and I found that I realized that it's really easy for the student to use it, so I'm happy also for that. But it's a powerful machine. Maybe you can have it with less. You have the one with a lot of laser, but we have just 4 laser, I guess, and basically we use 2. So we put the near ultraviolet because we want to see DAPI sample at microscope and flow cytometer. But in the end, we never did it. Then you get animated and say, let's do this, and you see the sample with DAPI and say, this look like a shit. I prefer cyber green, and so you just keep doing things as always. But yeah, I think it's a really good machine, this one.
Because I was thinking that maybe you using for regular routine control of microbial cultures that you do, and it's.
I think for regular things is you can use something like the Alcuri, because it's that you need the blue laser and the red laser, and that's it. And it's Alcuris look like a Lego machine. It look like Meccano. It's really it's light. You can move it wherever you want. We put on the boat. We put both, and they work it. Now the automatic sample they developed also for the Cytoflex. So we don't know, but we can use the Cytoflex in the lab because it's better. So
Antonio, please.
Besides the cost, I mean that well traded.
so the well-trained operator, it's me, and I mean, my boss, Pep Gasol, was really good in flow cytometer in the fax era. And I arrived to the lab after the pandemic, and the cytometer were into the box. And no one teach to me because, there was no. I have a little bit of training for the automatic sampler because it's complicated, but not for the cytometer itself. But I mean, you have just to read the manual. It's it's everything there. I think you, for example, you can understand how the machine work. They give you a little bit of training at the beginning. I didn't have it, but I've trained on other cytometer. So I think the machine more and more are user friendly. So because they want to sell it and they know that people want to do things fast, and I think Cytoflex is really, really easy to use, but it's like 100, 150. But I think flow cytometry, for this reason, I always say that the boss that are lazy send the student to flow cytometry, because you can always put two conditions and say, oh, more bacteria here, less bacteria here, because it's really easy. You can be a master, less than a master student, and use flow cytometry properly.
I have some questions related with these points. The price, more than the price, is the comfortability of using this equipment, as you mentioned, is becoming better. And my question is, there is a regular equipment, or it is possible to find one equipment that be able to help us to monitor the prevalence of eukaryotics, bacteria, eukaryotics, viruses and fungi, at least to explain, to have a look to the evolution of these populations. And then if we detect that in the cultures we have a crash or whatever, to be able to correlate and to say, oh, there was this tendency, and then to take samples and to send the samples to the omics guys in order to have a more detailed analysis of the.
Yeah, so I think for this kind of things, the automatic sampler is really, really good. And this works both with Akuri and Cytoflex. I don't sell it, but I'm really happy with the company because they really help you to each part of the things. And it's a little bit more complicated because you have to prepare solution and know that everything is in this place. But I mean, you can do it. It's not nothing.
But
and so about the groups, you can do normally. We did it. We did for now phyto bacteria. We also did the double staining every 50 minutes. What we don't know how to do it is eukaryotic that are not fluorescence, because I start to play with stains, but I have to see there are stains that are specific for flagella, for example, or stains that are specific for a component of the eukaryotic membrane. So you can say, oh, this is not bacteria, this is another thing. But I need time to do it. In general, no one do this because it's so easy to do it at we for example at our institute we have automated microscopy. So we use a probe for eukaryote, and it passed from sample to sample. So for now we didn't put effort. You can also use fish, but in this case it's more elaborate because there are people that do fish in cytometer. But in this case, I think the goal is to find something that is like cybergreen, but for eukaryote.
But what about viruses, etc.?
No, virus you can do it.
We can do that.
I think
the same equipment we can detect
Yeah.
using the
Yeah.
stains.
Yeah.
Okay. And the second question, because your experience in this type of samples, etc., in the natural samples, which is the damage of phytoplankton related with fungi or viruses? Because we always assume that fungi are the most problematic for microalgae. But in our collaboration in Prodigio, we detect a lot of viruses in the cultures, and we don't know what happened with the viruses.
Yeah, we'll actually start to study fungi now at the institute, so we have no expertise in flow cytometry. We use it a little bit of omics. And viruses, I think there are papers that show a correlation between the numbers from flow cytometry of phytoplankton and huge viruses. The problem with viruses that it's even more rough than bacteria because you really don't know what infects what. So it's more complicated. But there is a correlation. I mean, I think that yeah. I can pass you some paper later. About fungi, people not work a lot with that in flow cytometry for now. Actually, I add the stain because I think, oh, we never tried. I think yesterday we should try this to see if we can see fungi in natural environment, but there is a lot of work to be done.
I think the flow cytometry, as you say, is very valuable to explore this kind of interactions as a first step to be later in a deeper analysis using omics, because at the end omics must be the reference.
Yeah.
Yes.
The fact that it's omics, I think the big difference is that cytometry is fast and omics is not fast. And sometimes you don't have this time. And it's also a lot more expensive. So I think it depends on what you want. Maybe if you know that you always have a fungi things, you can start to see the fungi on your graph. Sometimes another interesting thing is that population have fluorescence in the wrong channel. You don't know why, but you can see population where shouldn't be there. Maybe you are lucky and your fungi is there. You can say, oh, when this dot appear here is because there is something. You never know. For this reason, I think you have to know your system to define population.
Yes. Antonio, please.
Yeah. Related to the experience that you had, you talked about environmental sampling. Yeah, but when you move to filtration, for example, algae filtration, do you see the differences in screening the biological established? For example, you had something highlights, I don't know, fungi, this kind of viruses, because we talk about this kind of project, science
I mean, I don't work a lot with culture, and if we work with culture, it's little things that we have in the lab. Sometimes you expect that your culture is really poor, and you see a lot of shit, and you can see when the culture is going down. And the people at my institute normally to monitor the culture fast, they use this. I have a friend that work with some Aptophyte. I don't remember the name of the things. And every day he just took a little sample and take a look because he want to be sure that he just take to the fluorescence, where if you have a fixed gate and all the parameters are the same, you expect to see your population always in the same position. So if you go down, there is less chlorophyll, and this means that maybe the health of the culture it changed for some reason. I don't know if I answered. You mean difference with the environment or difference in the
environment? Which are the highlights that you suggest to put it up?
I mean, if you highlight in the sense of environmental parameter or in the machine, I don't understand your question. Oh, well, I will always temperature and salinity. I mean, there is no oxygen, but
But I think we need to compare still the data from the reactor with the data from the natural environment, because we didn't compare yet this as part of the Prodigy project. We are only focusing in the influence of operational conditions in the evolution of the population. The problem and the advantage of the Prodigy project is that we have plenty of data. Thousands of samples, thousands of results from these samples, because we analyze the omics, the flow cytometry, the chemical fingerprint, the composition, the detailed composition of the supernata. The problem now is how to manage this information and how to combine. We are in this process now.
I think there is with flow cytometry, if the sample is too much complex, it's really difficult to understand what are you saying. You don't have a way. You can just sort the population and sequencing and say this is this. But of course, you cannot do this always. For this reason, if you have always the same closed system, you can understand more or less, and you know what is there. If it's an open system, it's a little bit more complicated. Yeah, but I tell because I saw it's prodigious, really complex sample. I don't know why, and they came from the reactors. There were a lot of things that appear, a lot of population. And normally I just do this cruise now, and for 3 weeks, battery move, but you have high and low from Vigo to Canaria. So it's really, well, it's like, I don't know, Anna was talking about this yesterday at the dinner, that, it's like a zoo, and you give a lot of food, and maybe the animal will be different that in the environment. I don't know.
Yeah, interesting though. I think it's not.
No way, we can talk later also.
This tool is about the screening. And so if you had a pattern during the time in your culture, it could be identified, for example, risky or a tendency during the time.
Yeah.
I think that it's not an easy work to do, and I think also Daniel wants the intention to having a pattern during the time.
Another way to do this is with the calculation of the diversity, because it's look like something that is ecological, but if you spot change in diversity in general, you don't need to know that this thing is changed, but something is changed, and you can check and say, oh, I will do omics now. And you can more or less automate this, I guess.
Thank you, Maximo. Thank you for all the speakers, and of course you for attending these lectures, and the people that also attend the lectures online. We finalize these formal lectures